Good morning, students. Myself, Major Komal Katyar, Department of Zoology, G.K. Govani Government College, Bhinmal. In continuation with my lectures on embryology, today we will discuss about the spermiogenesis, which is a part of spermatogenesis. It is also known as spermatoleiosis, which is basically the metamorphosis of immobile spermatid into a spermatozoon. Now, what is spermiogenesis? The spermiogenesis can be defined as a gradual differentiation of a stationary, rounded, and undifferentiated spermatid into an active and motile sperm. The spermatid formed as a result of second maturation division are typically undifferentiated animal cells and having only mitochondria, Golgi bodies, centrioles, but with a haploid set of chromosomes. In this form, it is incapable of functioning as a male gamete. During spermiogenesis, what happens? We can summarize them. First one is condensation of nucleus, or nucleus condenses. It means chromosomes condense and nuclear sap is removed. Second, the flagellum develops. Third, the spermatid elongates. Fourth one, the acrosome formed from Golgi bodies. The mitochondria aggregate around base of forming flagellum. The mitochondria fuse to form supermitochondria in the case of human beings, and most of the cytoplasm is shed and phagocytosed by Sertoli cells through tubulobulbar processes, and all these processes are summarized in the diagram. Now let us begin from the first, the nuclear changes. What type of nuclear changes occurs during the spermiogenesis? Apart from DNA, all other material of the nuclear sap are removed, including RNA and water. The chromosomes become closely packed. It means the condensation of chromosomes takes place. The shape of nucleus changes from spherical to elongated and narrow. It is an adaptation for propulsion in the liquid medium. The second changes, which are more important, formation of acrosome. Golgi complex is a spherical body, consists of numerous vesicles concentrically arranged cisternae in the spermatid, and they are replaced by one or two large vacuoles. Now, inside each large vacuole appears a small dense body which is known as the proacrosomal granule. If more than one vacuoles and proacrosomal granules are formed, these fuse together so that only one is left in the end. The vacuole with proacrosomal granule enlarges in size and moves to the anterior pole and gets attached to the anterior end of the nucleus, forming a sort of cap, and this is known as the cap stage of acrosome formation. Secondly, after enlargement of proacrosomal granules, it becomes acrosomal granule, which forms the core of the acrosome. This is known as the acrosomal stage. While the vacuole loses its liquid content and forms a double sheet over anterior half of the nucleus, and it is known as the acrosome cap. After the formation of acrosome cap, the remainder Golgi body undergoes gradual regression and is discarded as Golgi rest together with the cytoplasm of spermatid. Now next is the formation of axial filament. It is formed from the centrioles. Both the centrioles move and come to lie behind the nucleus. It means towards the posterior region of the nucleus, just opposite to the acrosomal region, one of which entered the depression in the posterior part of the nucleus and is known as proximal centriole, which we have already discussed when I told you about the structure of a sperm. It lies at the right angle of the main sperm axis. The second, or the distal centriole, lies near to it, but its axis coincides with the longitudinal axis of the spermatozoon, or we can say it is at the 90-degree angle with the axis of the proximal centriole. Next is the formation of mitochondrial spiral or mitochondrial axis. All the mitochondria from different parts of the spermatid accumulate in the middle piece of the spermatozoon around the proximal part of the axial filament and distal centriole. These finally form a spiral sheath around the proximal centriole, which is known as nimbus. Then the formation of manchidae. The manchidae, the cytoplasm of anterior region, moves to the middle piece and forms a condensed layer around its periphery, and this is known as manchidae or cytoplasmic droplet. All the stages we have discussed so far are summarized here in this slide. The first, the spermatid nucleus, and there is development of acrosome cap, which is elongating, as well as the spermatid nucleus is also undergoing the change in its shape. There is formation or development of flagellum. Shape of spermatid starts changing. The axis of cytoplasm is removed. The mitochondria are so we are talking about the mitochondria. The mitochondria, which were initially segregated, start collecting at a particular place, and then they are mainly present only in the middle piece. In the last diagram, it is clearly visible that there is an acrosome cap. The head region is clearly visible with nucleus, then the middle piece with the mitochondrial spiral, and the tail or the flagellum. So the spermatids are transformed into sperms, also called as spermatozoa, by the process called spermiogenesis is depicted here. And after the spermiogenesis, sperm cells become embedded in the Sertoli cells and are released from the seminiferous tubules by the process called spermiation. So here the two terms are used. One, the spermiogenesis, which is the metamorphosis of spermatids into sperms or spermatozoa, and the second one is the releasing of sperms from the seminiferous tubules, which is known as the spermiation. Now, what are the function of Sertoli cells? They remove the excess cytoplasm from developing spermatid, which is by the tubulo-bulbar processes. Second, they are responsible move spermatids towards the lumen of the seminiferous tubules, which is the ectoplasmic specialization. They are responsible for nurturing and mediating the maturation of spermatids. They segregate the groups of developing gametes. They are responsible for secreting fluid to transport sperm into reproductive tract. They are responsible for secreting hormones and other factors. For example, in the embryonic stage, they are responsible for releasing anti-Mullerian hormone. In adult, they are responsible for releasing inhibin, which inhibits the FSH production, that is follicle stimulating hormone. Estrogen may act to inhibit GnRH, the gonadotropin releasing hormone production by basal hypothalamus, and other factors which are not hormone, the androgen binding protein, which helps to transport androgens from interstitial fluid into seminiferous tubules. It promotes the process of spermatogenesis. Now let us compare the spermatogenesis and spermiogenesis. The spermatogenesis is the formation of motile spermatozoa from spermatogonium, while spermiogenesis is the production of mature spermatozoa from spermatids. 4 functional spermatozoa are produced from one spermatogonium, while a single spermatozoon is formed from a single spermatid. The spermatogenesis consists of 3 phases: the multiplication phase, the growth phase, the maturation phase, and a differentiation phase, while the spermiogenesis consists of only a differentiation process. The spermatogenesis is a whole process of production of mature sperm cells from the primordial germ cells, or PGC, while the spermiogenesis is the final differentiation phase of spermatogenesis. It means it is a part of spermatogenesis where mature spermatozoa are produced. In the case of spermatogenesis, the diploid germ cells become haploid gametes, while in the case of spermiogenesis, it occurs only in the haploid cells and there is no change in the amount of genetic material. The number of cells that are capable of differentiating in spermatozoa are increased, and in the case of spermiogenesis there is no change in number of cell occurs because only the differentiation takes place during the spermiogenesis. Now, what are the significance? The haploid male gametes are formed. Second, 4 haploid spermatids are formed. The non-motile spermatid changes into motile sperm, that is, we have already discussed today the process of spermiogenesis, and spermatogenesis is a process of maintaining species specific chromosomal number constant. So these are the 4 significance of male gametogenesis or spermatogenesis. Now there are 2 types of breeders. One, the non-seasonal breeders, and the other one are the seasonal breeders. In the case of non-seasonal breeders, the act of reproduction continues uninterrupted. It means they breed throughout the year. So in them, the spermatogenesis is a continuous process. As a result, the sperms are produced throughout the reproductive span of the male. It means the male is always fertile, as in the case of pig or in the case of cattle, which are polyestrous. The second case of seasonal breeders. In the case of seasonal breeders, there is a testicular cycle similar to the ovarian cycle, and the sperms are produced only when female is ready to receive them. The testicular cycle is differentiated into 4 phases. For example, horse, sheep, and goat, which are monoestrous. And the 4 phases include a breeding phase, a regressive phase, a refractory phase, and a progressive phase. So this finishes my lecture on spermiogenesis. Thank you.